welcome to Virtual Investor Conferences. On behalf of OTC Markets, we are very pleased you've joined us for the Life Sciences Investor Forum. Our next presentation of the day is from Defense Therapeutics. Please note you may submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings by clicking Book a Meeting in the top toolbar. At this point, I am very pleased to welcome Amy Finney, Director of Strategy and Business of Defense Therapeutics, which trades on the OTCQB venture market under the symbol DTCFF and on CSE under the symbol DTC. Welcome, Amy.
Thank you very much. It is my pleasure to introduce all of you to Defense Therapeutics. We are a Canadian company that aims to become a leading drug delivery platform company. I thought I would start off today sharing a little bit about our journey over the last 5 years, growing this exciting company around this exciting technology. Defense Therapeutics was launched in 2020 after the licensing of the ACUM drug delivery platform from Sherbrooke University professors in Quebec, Canada. Like most biotech companies in the early 2020s, we immediately jumped into the vaccine world to assess how our technology could support the vaccine efforts of the world during the pandemic. We made some exciting progress there and have those products in our pipeline continuing to develop today. On the heels of that, Defense Therapeutics expanded from the university into an independent R&D laboratory at Montreal Innovation Park. Here, we have continued to develop an exciting pipeline of products, including ACUTOX, which has been approved for clinical trials for melanoma. Over the last few years, we've expanded our R&D efforts to investigate how ACUM, this drug delivery platform, can impact other biologics, most notably ADCs and radiopharmaceutics, areas which are very exciting in the drug delivery and oncology space today. We are incredibly excited about the results that have been generated over the past few years, and I'm going to share some of those with you today. And because of this, we've set out a strategic plan around those two programs, expanding from 2025 to 2027. And I'm going to share a little bit about that with you today. The three programs that I want to talk about today are our 2026 value inflection programs. The first is our ACUM ADC program. The second is our ACUM radionucleotide drug conjugate program. And the third will be our efforts to manufacture ACUM products for both ourselves and our partners. So let's start on the ADC platform. For those of you who are unfamiliar, ADCs are antibody drug conjugates. These are drugs that entered the R&D field about 25 years ago as a response for precision delivery of potent chemotherapy agents. How I like to describe these drugs is a carrier for most potent drugs. So most people know that chemotherapy patients who receive chemotherapies often get sick because the chemotherapy agents are delivered to the entire body. When antibodies are used to carry these high potent cytotoxic payloads, you get a precision delivery directly to the cancer cells. And these ADCs have proven to be an exciting next generation of precision oncology drugs. However, the promise versus reality is quite different. ADCs that are on the market right now are mostly approved as second-line therapies, and those in clinical trials are continuing to fail. Although ADCs are certainly delivering on the promise of precision tumor targeting, they find the tumor cells very well, they can carry high potency loads, their use and development is limited by intracellular payload delivery challenges. As you can see here on the screen, once the ADC enters the cell, it gets encapsulated in an endosome and has a lot of trouble getting out of those endosomes. And in fact, some of the literature suggests fewer than 5% of the antibody drug conjugates that are given to the patient actually escape that endosome and are able to do the killing job inside of the cell. Because of this entrapment and this delivery problem, there's a narrower therapeutic window than was hypothesized with ADCs. High doses are required for patients in order to have effective killing of the cancer cells. This leads to, unfortunately, a lot of side effects and toxicities associated with these drugs. Now, because of this intracellular delivery challenge, a lot of ADCs are failing to deliver their potentials. On the market and in development, drugs are struggling. Here's just a few of the statistics. Of the 20 or so ADCs that are on the market for oncology, 19 of them are only second-line therapies because they know that they have these toxicities associated with them. Also, unfortunately, because of these toxicities, approximately 40% of patients who do receive these drugs don't complete the regime of therapy because of the toxicities and side effects that they experience. That 40% number pops up again when you look at the clinical trials that are currently being stopped due to safety concerns. And ADCs, because their potential is so high, there's more than 1,000 active trials right now in the USA alone for different ADC drugs. So that kind of failure rate has significant economic and patient impact. So what if a molecular tag could be added to ADCs? And I've represented that molecular tag with our two little glow-in-the-dark cartoons here on this diagram. And that molecular tag could help facilitate escape from the cell's defenses, escape from that endosome that was on that first diagram, and also help drive the payload to the center of the cell, to the cell's nucleus, where it needs to be in order to effectively kill the cancer cell. If such a tag could be added to overcome these hurdles that ADCs are challenged with, we could have expanded therapeutic windows and lower doses would be needed to achieve therapeutic effects. This is exactly what the ACUM innovative drug delivery platform can do. ACUM is a molecule that is a combination of two naturally occurring intracellular trafficking molecules. One, a cholic acid moiety, which facilitates endosomal escape. The other, a peptide signal, which is a nuclear localization signal used by different biologics to naturally navigate intracellular trafficking. This molecular tag has been specifically engineered together, and it facilitates, when it's put together, it facilitates the intracellular accumulation that improves precision drug delivery. Now, Defense Therapeutics has 8 granted patents protecting this drug delivery platform and its use with a broad variety of biologic drugs. I want to just show you some of the exciting data on our ADC program, though. So one of the early first generation ADC drugs is for HER2 positive breast cancer, and the name of the drug is Kadcyla. It is a Genentech drug, and it is been on the market as a second-line therapy for HER2 positive breast cancer for more than 10 years now. This drug offers us a great opportunity to study the effects of ACUM enhancements because of how well characterized it is. So I'm going to show you some of that data now. Our scientists attached ACUM molecules to Kadcyla in 3 different loads, if you will. A low AAR, a low ACUM antibody ratio, a medium antibody ACUM ratio, and a high ratio. And what you can see is that this laboratory model of cell survival, these are HER2 positive breast cancer cells from a patient that have been cultured in a dish, and then the drug has been added. You can see the black line. Kadcyla is actually an effective killer of these cells. That at low doses they survive, and as the dose increases, you can see the survival goes down. When we add ACUM to this drug, there's a significant shift in the potency curve of Kadcyla, some ranging between 50 and even up to 100-fold, depending upon the load of the drug, of the ACUM on the Kadcyla. So it was this kind of data which really excited our scientific and business team, and we really started to realize the potential of how ACUM can enhance the potency. The next set of data we went after, of course, is in vivo proof of concept. And here we use mouse models of patient-derived xenograft models of HER2-positive breast cancer, again, in combination with Kadcyla. And the results were equally as exciting to our team. You can see here, again in black, the tumors that are injected into the mice triple in size within about 3 weeks if there is no treatment. With a low, non-effective dose of Kadcyla, the tumors continue to grow as they did with no treatment. We upped the dose of Kadcyla a little bit. This was chosen because it's a non-toxic dose. It's a sub-effective dose. As you can see, the tumor still grows. However, you've got a little bit of room for improvement there. Then we took ACUM and added it to the ADC and gave it at that non-effective dose, that 0.5 milligrams per kilogram dose. And you can see in green how spectacularly ACUM enhances the efficacy of Kadcyla. How is it doing this? Just as we predicted. Here is a few pictures of some of those HER2-positive breast cancer cells that have been cultured in our lab, and we have labeled the Kadcyla with red here to show its location inside of the cell. Now the ring is the outside of the cell, and the blue is the nucleus of the cell. You can see Kadcyla alone tends to stay on the outside of that cell. But when we tag that Kadcyla with ACUM, the cell gets fully penetrated. The accumulation of the Kadcyla inside of the cell is quite spectacular and even tends to concentrate at the nucleus. These results with Kadcyla were incredibly exciting, but our excitement grew when we saw that this was true for not just the HER2-positive breast cancer cell lines, but across multiple different cell lines, HER2-positive cell lines, and more so that we've used now novel ADCs that are ineffective at an ineffective dose, such as this one here. Again, the ADC here is a novel ADC. Tumor volume is growing as expected, and the drug on its own is doing nothing. That same dose of that ADC, when we tag it with ACUM, flatlines that tumor growth. Really exciting for all of us. It was these results which convinced us that the potential impact of ACUM on the ADC market needs to be our strategic priority. The ADC market is booming, but there remains a major unmet need. ADCs continue to be challenged by payload delivery problems, and the ACUM drug delivery platform directly solves the #1 failure point in ADCs right now. Our platform has a broad plug-and-play enhancement applicability that has the potential to impact all ADCs across all fields. Because of this platform potential, we have the opportunity to have broad partnerships for impact across all the drugs that are on the market, in development, and even generic drugs. We've got multiple shots on goal to generate revenue through co-development deals, licensing revenue, and even trying to leverage the exciting field right now where there's a lot of acquisition interest from companies that are racing to differentiate their ADC pipelines. And in 2026, we are going to go after all of these business prerogatives. We have a robust partnership and licensing strategy that we are ready to launch. We have pharma and biotech partnerships that we are exploring, discussing with different groups about how to revive franchises which are coming up for patent expiries, how we could shift second line to first line therapies, and very excitingly to our potential partners, how we could potentially rescue some of those late stage failed ADCs. We're also in discussions with biosimilar groups. As I mentioned, ADCs have been on the market for a while. Some of the patents are expiring in ADCs, and biosimilar generic companies are scaling up biosimilars. We have started some discussions with those groups to see if we could co-develop new patentable ACUM enhanced ADCs with them. Of course, an incredibly robust area for us for potential partnerships is with startups and early SMEs who have novel ADCs that are having some of these delivery challenges. And we hope to license and partner with a lot of these companies across the globe. Finally, we have a partnership established with a company that has a linker technology, which we are hoping to be able to leverage as a combined product to enhance ADCs in a plug and play options. 2026 is going to be an exciting year for our business development team and for our laboratory team. Before I get into a little bit of that, I just wanted to show just a little bit of the data coming out of the markets, which really convinced us that 2026 should be the year that we are really going after this strategic priority. If you take a look at the different deals that have been ongoing over the past couple of years, it's quite clear that there's an outright land grab for ADC enabling technologies by the major pharmaceutical companies and biotech groups that are developing ADCs. These are just three examples out of 2024 and 2025 deal analysis, which show these deals, they're in the billions of dollars. And so we are really excited to try to leverage some of this momentum in the market for our ACUM drug delivery platform. This is also just a quick example I wanted to add because I think it's really important to understand the difference between being a second-line therapy and being a first-line therapy. Having such toxicities due to that narrow therapeutic window and keeping ADCs as second-line therapies has significant impact for both the patients who need these drugs the most and the companies that are creating the ADCs. Second-line therapies such as Kadcyla and HER2, although having quite a generous revenue streams annually, about ranging between $2 to $4 billion. Those first-line therapies are over $5 billion. So shifting from a second line to a first-line therapy has significant impact for patients and manufacturers. Okay, before I go on to tell a little bit about our manufacturing strategy and what we're doing in our R&D lab in the Innovation Center, I want to introduce our second program. Because if there's anything hotter than ADCs right now in the life sciences market, it's radiopharmaceuticals. We have 2 radiopharmaceutical programs active, one an in-house development, which is 100% owned by Defense Therapeutics, and the other a partnership program. Why are we excited about radiopharmaceutics? It comes back down to the biology. In development, we know that there is radioactive isotopes can be used as the payload associated with antibodies. So again, leaning into the precision delivery offered by antibodies very specifically to cancer cells, the cytotoxic payload can be radiation. This is a new and exciting field which is growing so fast in the life sciences market. All the major pharmaceutical companies, again, are going out into the early stage companies and looking for technologies that can enable this. What's the big challenge faced with these kinds of drugs? What's going to stop them? Very similar to ADCs carrying chemotherapies. These drugs need to get inside of the cell, escape the cell defense systems, and get as close to the nucleus as they can in order to make their effects, their killing effects. This is even more important if you've got a radioisotope. With these new alpha emitters, the proximity, they have such a small radioactive emitting diameter that they really do need to be at or on the nucleus in order to kill the cell. We are so excited because this is exactly what ACUM does. ACUM sends payloads directly to the cell nucleus. You can see it in our micrographs. And so we have collaborated with the Canadian Nuclear Laboratory here in Canada to be our collaboration partners and develop independent novel drugs in the radiopharmaceutical space. We're really excited. In 2026 and Q1 2026, we are expecting the first proof of principle results to come out of those experiments, and we'll be very excited to share them with the field at some of the upcoming conferences in 2026. All right, so those are our 2 programs. We've got the ADC program and we've got our ARC program. Both were excited about the progression of our own internal R&D results, as well as our partnership efforts. Now, what we are doing in-house at this moment to ensure that our business development efforts and our partnership efforts are going to progress as fast as possible is we have set up a validation program that will validate and optimize ACUM on antibody-delivered drugs for any of our partners' drugs. We've got 6 stages. We've got each of these stages well validated and documented in our laboratories so that we can have a very rapid, thorough screening, validation, and optimization of the impact of adding ACUM to anybody's antibody-delivered drug. We're really excited about that laboratory build-out. And it feeds directly into the last part of our strategy, which is our manufacturing strategy. We have initiated the establishment of an in-house quality control laboratory to ensure the quality supply of ACUM for our own programs as well as for our partner programs. Our aim is to have a drug master file as a drug additive filed with the regulatory groups by the end of 2026, so we can truly offer a plug and play drug additive for our partners. We're modeling this strategy after Seattle Genetics, or Seagen's dual revenue strategy that they were so successful on when they began their adventures as a drug linker company. And they were able to do both licensing and supply for their customers and their partners. Anybody who follows the industry likely is familiar with that story, because the acquisition of Seagen following the successful implementation of that strategy led to their 2023 acquisition by Pfizer at one of the highest values in biotech history. So just to close up, I'd like to just remind everybody about our 2026 value inflection year. We have it well mapped out strategy in which we are going to be able to extend our current partnerships with public announcements. We are confident to be able to close and to finalize some of the biotech and pharma partnerships we're currently in discussion with by mid-year. And we are very excited that we should have a candidate identified for preclinical development in our internal radiopharmaceutical research by the third quarter. And finally, that filing of the DMF and ready for first sales to our partners by the end of the year. So just wrapping up now, a little bit again about our leadership. We're an experienced team, myself in strategy and business with 20 years experience in pharma and biotech across Europe, USA, and Canada. Sebastian, who's going to join me in a few minutes for the Q&A, is our CEO and founder and has 30 years of entrepreneurial experience in publicly listed companies. And Sarkis, who is our medical director and is a world-renowned oncologist and is leading up a lot of our clinical strategies at McGill University in Montreal. I think I'll close up there. I will close up there and we'll get ready for questions. Because we have a call to action for all of you to join us now. We are currently have a task open of raising up to $10 million in equity financing that we are aiming to close by the end of January, with the use of funds focusing on that R&D capacity to make sure that we can validate and optimize ACUM on anybody's ADC that wants to partner with us, scale up that business development effort, and of course, prepare those regulatory filings and manufacturing for ACUM sales to start by the end of the year. I think I'll leave it there. And in the last few minutes, we'll invite Sebastian to join me and we can take some of the questions from the audience. Sebastian?
Yeah, Amy.
You got a question for us?
Yes, thank you. Thank you very much for your great presentation. That was very well done. So Amy, we have a few questions from the audience that I'd like to read you. So let's start with one here. So given the recent news flow and increased BD activity, what are the top two catalysts in the next 6 to 12 months that you believe could meaningfully change how institutional investors value defense?
So I think the expansion. So as we've said, we've got two active partnerships right now. One with a company who is doing some R&D and some validation work with us, and we hope in Q1 to be able to publicly announce the expansion of that partnership into a licensing deal. The second probably comes with the radiopharmaceutical data that is being generated right now. Again, two active programs there. Both are going to read out in vivo proof of concept data in Q1. And so we're hoping that that will also trigger that partnership, the one program that is partnered to become public, and also be able to make public the lead asset that is under Defense Therapeutics control ourselves. So we've got an exciting Q1 that is coming up with a lot of data, and hopefully a lot of partnership announcements.
Yeah, thank you. Here's another question. So any ADC companies can potentially reduce the dosage and have the same efficacy by only adding your ACUM molecule, meaning less toxicity for the patients. Do we have toxicity data from ACUM and or ACUM plus ADCs?
Yes, right. Our data is supporting the idea. You know, we're getting more and more data from our R&D team, literally daily, to suggest that when we tag an ADC with ACUM, that increases the potency, right? And it's really just a matter of optimizing how many molecules of ACUM need to be added to the ADC to keep all of the PK properties intact and to increase the potency. So it's a little bit of a balancing act, and that's why we're establishing the in-house laboratory. We know how to do this, and we're gonna drive that forward. And so it's an exciting opportunity for a lot of different ADCs.
Great. So maybe we have time for last one. So following your meetings at the World ADC and CPHI, how many distinct ADC or radiopharma companies are now actively evaluating ACUM, and what are the next concrete steps to convert those into formal deals?